Hello, this is the explanation, part one of mitosis. Mitosis. Sorry, I messed up here. I tried to cover it up with whiteout, but it didn't work. So, mitosis is a type of cell division that results in two daughter cells having the same number and kind of chromosome as the parent nucleus. The Google Dictionary definition. These are the phases. First phase is interphase. The cell spends most of its life in this phase. In this phase, the cell copies its DNA in preparation for mitosis. This is the happy cell. Chromosomes are right there. Second phase is called prophase. This is the process that separates the duplicated genetic material into two identical daughter cells. Twins! There you go. The third phase is called metaphase. The duplicated chromosomes line up in the middle of the cell before separation begins, as you can see right here.
In anaphase of mitosis, the sister chromatids separate from each other and are pulled apart towards opposite ends of the cell. The protein glue that holds them together is broken down and allows them to separate. Now, each is their own chromosome, and the chromosomes of each pair are pulled towards opposite ends of the cell. The microtubules, non-attached to chromosomes, elongate and push apart, separating the poles and making the cell longer. In telophase, the cell is nearly done dividing and starts to reestablish its normal cell structure during cytokinesis, the division of cell contents. The mitotic spindle is broken down to its building blocks, and new nuclei are formed, one for each set of chromosomes. And the nuclear membrane and nucleoli also reappear. Now, for meiosis, before meiosis can occur, cell must go through interphase, and as in mitosis, the cell grows during G1 phase, copies off its chromosomes during S phase, and then prepares for division in G2 phase. During prophase, the differences from mitosis and meiosis begin to appear. As in mitosis, the chromosome begins to condense, but in meiosis, they also pair up. Each chromosome carefully aligns with its homologue partner so that the two match up at the horizontal position along their full length.
So for my section of the presentation, I'm going to be covering some of the end parts of meiosis as well as crossing over. The first part, metaphase 1, that is the part where the nuclear membrane disappears. The microtubules form into a spindle, and the fibers and homologous chromosomes begin to separate to two sides of the cell. So that's something that we looked at in class under the microscope. Additionally, in anaphase 1, that's when the two homologous chromosomes separate to the actual complete sides of the cell. The fibers shorten as well, and the pulling of the chromosomes towards the side of the cell occurs. So that's kind of where you had these little like X's, and they start just getting they have the fibers in between. The X's are being pulled off on either side of the fibers. So the next part, telophase 1, that's the final stage of meiosis, and that's where the chromosomes are completely pulled to the opposite sides of the cell. The previous phase, anaphase, that's where they were beginning to be pulled apart. Telophase, that pulling is essentially completed. The nuclear membranes do reform to produce two separate nuclei. So the next thing that this presentation covers is crossing over. Crossing over is interesting because that's where pretty much genetic material is exchanged in between two homologous chromosomes. So essentially when a mom and a dad get together, create a child, that's why the child has the traits of both, not just the traits of the father or the mother, because just that sharing of genetic material.
Cells move from meiosis 1 to meiosis 2 without copying their DNA. Meiosis 2 is a shorter and simpler process than meiosis 1. It may be helpful to think of it as mitosis for haploid cells. And during prophase 2, chromosomes condense and the nuclear envelope breaks down if needed. The centrosomes move apart and the spindle forms between them. And the spindle microtubules begin to capture chromosomes. The two sister chromatins of each chromosome are captured by microtubules from opposite spindle poles. In metaphase 2, the chromosomes line up individually along the metaphase plate. In anaphase, the sister chromatids separate and are pulled towards the opposite ends of the cell. In telophase 2, the nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final products of meiosis, four haploid cells in which each chromosome has just one chromatid.
One key difference between meiosis and mitosis is meiosis is used for the production of body cells, like tissue or cardiac cells, whereas meiosis is used to produce sex cells, also called gametes or germ cells. Mitosis produces 2 identical daughter cells. Meiosis produces 4 unique cells.